This is a production of Cornell University.
So, hello everybody, and thank you very much for being here. I know it's very cold and it's early in the morning, so I really appreciate it. And so, as Susan told you recently, I used to work in Uruguay at the university, and I was going to college there, and I had just finished my master's in which I did my master in genetics and mechanisms of genetic tolerance. Mechanisms for controlling cold tolerance in rice. So I had just finished my master, and I was looking for a PhD, and I was collaborating very closely with the Uruguayan rice breeding program at that time. I never worked for the Uruguayan breeding program, but I used to collaborate a lot and still do. So I was looking for a PhD, as I said, and so the Uruguayan breeding program is a small and traditional breeding program. So what they were trying to do, they were trying to start to thinking about how to implement genomic selection in the breeding program. And since I was around, I took that task, and I decided to base my PhD project in this. So what I try to do is to explore some of the aspects related to implementing genomic selection in the rice breeding program. And I don't know if I did the best choice, but that's how I got here. So first of all, where is Uruguay? Uruguay is this very tiny little country here in southern South America. And as you can see here in this map, it's right in the limit between the subtropical and temperate zone. So for this reason, the Uruguayan climate can be classified as humid subtropical. So we have 4 very well-defined seasons. And so the most extreme, of course, are winter and summer. And the winter runs, the winter runs from June to August, where the average temperature can go between 3 to 10 degrees. And then we have the summer season that runs from December to March, where the average temperatures can be between 17 to 28 degrees. So for these reasons, rice cycle usually takes over during this period. And since temperatures during the winter are so low for rice, we can only have 1 cycle of rice a year. So then we usually don't tend to associate Uruguay and rice, but actually rice is neither Uruguayans do. But rice is the 3rd top export for Uruguay. Uruguay is mainly an agricultural country. So rice comes right after beef and soybean. And also Uruguay is among the top 10 major rice exporters in the world. And it's actually on the 1st place in Latin America. And 95% of the rice production is exported. So we're not a rice consuming country. And for this reason, we have to meet very high standards for grain quality to be able to export our product in the international market. And also, as you can see here in this plot, Uruguayan rice yield are also very high. They're between 8 to 10 tons per hectare. And as we can see here in the plot, this is the progression of rice yield since 1970s, where the rice breeding program was firstly established in the country. And we can see that how the grain yields were increasing over time. And this was thanks to the adoption of new high-yielding varieties, but also to the investment and the adoption of new agricultural practices, and also a lot of machinery investment. However, no. So as I said, the high-yielding varieties from Uruguay were developed by the INEA, which is the National Agricultural Research Institute in Uruguay, and the Uruguayan rice breeding programs belongs to INEA. And this breeding program consists mainly of, is divided in 2 main breeding programs, which is the tropical japonica breeding program and the indica breeding program. The tropical japonica consists mainly of lines that were introduced from the US in the 70s, and they were adapted to Uruguayan conditions. And the indica germplasm consists mainly of varieties that were introduced from IRRI in the Philippines and SIAT in Colombia. And there's only three varieties that were released by INEA account for 92% of the planted rice area in Uruguay. So these main three varieties are El Paso 144 and Olimar, which are indica varieties. And then there's a tropical japonica variety, which is INEA Taquari. So the main problem we're facing right now is that these varieties are reaching their maximum yield potential. So and this was thanks to all the improvements we did in the agricultural practices and machinery. So we're really in the urge to develop new varieties with higher yield potential. But also yield is not everything in rice. Grain quality is very important too. And since we export most of our production, we really need to meet those high standards in international markets. So the main components of physical quality are length and width, which determine which kind of grain we're having, either short, medium, or long grain. And then head rice yield is a very important component of grain quality because head rice yield is the weight of whole kernels, because nobody likes broken kernels. So this is very important, and it really determines the price of the rice bag in the market. And then color is important, and also chalk is another very important trait. And chalk, because chalk really induces grain breakage, so it reduces head rice yields. And chalk is this opaque area in the grains that is caused by misfolding of the amylose and amylopectin layers during grain filling. So that was this opaque area induces grain breakage. So this is a very important trait too. And cracks are also important because they also induce breakage. So we know that milling quality traits and yield are the traits I'm focusing here in my project. So, and we know these traits are quantitative traits. So genomic selection could be a good strategy to improve these traits. And that's what basically my project was to explore some of the aspects of implementing genomic selection to improve both grain and milling quality traits. So particularly, I focused on evaluated the impact of accounting for GWAS in our prediction models. And then also in the GWAS area, we compare different ways to fit weather covariates to feed our models and improve our prediction accuracies. Also, another thing it was the use genomic selection to select the best combinations of parents to cross and start new breeding cycles. And this is like a bonus track of the thesis, which is finding genomic regions that are associated to anther-cultural response when we try to develop double haploid lines for breeding. So the first thing I'm going to talk to you about is to evaluate how to evaluate prediction accuracies for yield and milling quality when we account for GWAS in our germplasm. And this is part of a work we published in Crop Science last year. So there's a lot of different ways to account for GWAS statistically. We were focused mainly on using mixed models. And so the mixed model we used had this general form where we have a trait, our response variable, which is the measurements of the traits for all genotypes in all environments. Then we have the mean of the environment. And we have this genetic component, which is the random genetic effects. And these genetic effects have a normal distribution with this covariance structure, which can be modeled by the Kronecker product between two different components. One environmental component, which is this UE matrix here, that represents the genetic covariance between environments. And this K is the genetic covariance between lines. That's the genetic component of the model. And there's a lot of structures for these genetic covariance between environments. So we were focused only in two main structures, which are actually they're kind of like opposite, which is the diagonal matrix and the unstructured matrix. So the diagonal matrix, what it does, it fits one single variance per environment, and it treats, and then there's no covariance between environments. So by this way, we're accounting is the same as fitting environments or treating them as independent. And we're not sharing information between environments. Then the unstructured covariance matrix is as the diagonal matrix. It fits one single variance per environment, but it also fits a covariance between pairs of environments. So we try to compare our prediction accuracies with these two methods. The data sets we used were tropical japonica and an indica data set that consisted both of 300 elite breeding lines. And these lines were evaluated in 3 different years. And the traits we used were yield, milling yield measured as in kilograms per hectare, then milling yield, head rise percentage, and grain chalkiness. These are the 3 main components of milling quality and plant height. I'm going to explain now how we phenotype these traits because we're going to refer to them along the whole presentation. So when we first get our harvest our rice from the field, we get the paddy rice or rough rice, which is the grains that they're still attached to the hull. So we have to dehull these grains to obtain the brown rice. This brown rice will go then will pass through the mill, and it will be to obtain the white rice. Then after milling, we're going to obtain our white rice, which is it constitutes of both whole kernels and also broken kernels. So the total amount of both whole and broken kernels is the milling yield relative to the original amount of rice, right? Then the head rice is only when we separate both broken from whole kernels is the total amount of whole kernels relative to the original amount of rice. And then chalkiness is the third component, which is measured as total chalky rice over the total milled rice. And chalky grain is grain is considered as chalky, when the chalky area is more than 50% of the total grain area. So we used several cross-validation schemes. Here I'm going to show you just a few of them. So we used a cross-validation 1 and 2, where the cross-validation 1 is tries to predict lines that were never evaluated in any environment. So they're new lines. And the cross-validation 2 predicts lines that were evaluated in some environments, but not in others. So here we're really using the information that runs through the environmental covariance matrix. And these are the results from the prediction accuracies for each trait in the indica data set. And I know there's a lot of things going on in this plot, but I want you to focus only on these 2 pink bars for each year and each trait. These two pink bars correspond to the cross-validation scheme 2, the one when we try to predict lines that were tested in some environments, but not in others, using the unstructured covariance matrix. So then these are actually the only ones that are kind of like more different than the rest. And for tropical japonica, we see kind of the same trend, but not as pronounced as with the indicas. Again, using the cross-validation 2 with the unstructured covariance matrix, we can achieve the better prediction accuracies. And another cross-validation scenario we tested, and I'm going to tell you, want to talk about today, is when we're trying to predict new environments, so environments that were never evaluated before, right? In our case, since environments are years, we're trying to predict future years, using the information from previous years and the training population. So here we can see when predicting each year for both the indica and the tropical japonica for each trait, using the diagonal and the unstructured covariance matrix. And we can see that in each case, prediction accuracy is a little higher when using the unstructured covariance matrix than the diagonal matrix. So actually using fitting these kinds of models that allow us to borrow information between environments really help us to predict our lines, to achieve better prediction accuracies. So as a summary of this part, again, the unstructured models, when you fit in unstructured covariance matrices, really usually under the cross-validation 2 scenario, give better prediction accuracies than when using diagonal covariance matrices. And also using these types of matrices are, are, um, are beneficial when we try to predict untested years. And also this is also true because in the case of the unstructured covariant matrix, the correlations or covariances between environments were very high. So for these reasons, those high correlations really improve our prediction accuracies with these types of matrices. And this basically responds to the stable weather conditions in Uruguay where there's no major weather events. So usually, I mean, weather is stable, there's still variation, but there's no big variation, right? So using models that account for G/E and that account for correlations between environments can be beneficial to predict these traits. So now this is another study where we also use G/E models, but here what we wanted to do was to feed our models with weather data to be able to model our environments in a better way. So the objectives here were to explore different ways to include these weather covariates in our models, because the problem that we have when we use weather covariates can be similar than when we use molecular markers. Weather covariates can be too many and they can be correlated. They can be highly correlated between each other. So it's not easy to fit these types of models. And also another objective was to try to study the genetic basis of G/E in our own germplasm. So the environmental covariates we used they were retrieved from the weather station in the rice breeding program, like right when where the lines were evaluated in the same field. And they consisted of either temperature, solar radiation, precipitation, and they were daily measurements for all these different components. And basically, we know that these covariates, they're not going to affect on the same way in each state of the growth, of the plant growth. So we try to calculate, so we try to short those covariates to different stages so we can model it more accurately. So we try to divide our plants, the rice growth into three main different phenological stages, which were vegetative, reproductive, and maturity. Reproductive is the flowering, and maturity is grain filling stage. So the problem is that we didn't have the information of what time in the field plants were at each stage. So what we used to calculate these stages was the flowering time. And we know usually the flowering stage from budding to flowering takes about 35 days. So based on these measurements, and we know that plants are left in the field after flowering for about 30 days for grain filling, and knowing the date of planting and of harvesting, we can calculate these stages for our plants in the field. So we calculated our covariates as their averages over all these stages for both the tropical japonica and the indica data sets. And here the data sets we used were the same as in the previous work. The only difference was that the tropical japonica data set had 2 more years of evaluation. And the traits used were grain yield, head rise percentage, chalkiness, and height, plant height, as a control for a trait that is not controlled by the environment so much. And we try to compare two main different types of statistical models here. So we have on one side the reaction norm models that were developed by Diego Jarquin, and the PLSR models, which PLSR stands for partial least square regression models. So for the reaction norm models, we tried 3 different models, which one that only models the genetic effects that were modeled using, these effects are random, so they were modeled using the covariance matrix between genotypes. Then we have a G+W model that has a genotype component and an environment component, and the environment component was modeled using the environmental covariance. Sorry, the matrix of environmental covariates. Sorry, the matrix of covariance between environmental covariates. And the G+W+GW model adds this interaction term between genotypes and an environment, and this term is modeled by using the multiplicative product between the two relationship matrices. On the other hand, we have the PLSR models, and basically what PLSR models do, they decompose our data into latent variables, and these latent variables are linear combinations of our data. And what they try to do is to maximize the covariance between our response and our predictors. So here we try the PLSG model, which was only using markers as predictors, and the PLS-GW model, using both markers and environmental covariates. And these are the results for the, oh, sorry, I don't know why they don't show up here. But these are the results for the prediction accuracies in both data sets. And here in blue, to predict each year. So here we're trying to predict new years. So the blue bars here represent the reaction norm models. And the red and orange bars represent the PLS models. So we can see that for the indicas and also for the japonicas, but mostly for the indicas, the PLSR models gave better prediction accuracies or equal prediction accuracies than the reaction norms. But usually this orange bar, which corresponds to the PLS using both molecular markers and environmental covariates, they usually give a little higher prediction accuracies. We can see it here too for the japonicas. So basically, for these data sets, we obtain better prediction accuracies when using these types of models, the PLSR models. Also, as I said in the introduction, we try to explore the genetic basis of GWAS from our germplasm. And what we did was we perform a GWAS for each year, each trait in each environment. Each year. And we could only here we could only find significant QTLs for head rise and chalkiness in both data sets. We couldn't find any significant QTLs in any environment for grain yield. So what we did here, so all these QTLs have different effects in different environments, right? So what we tried to do here was to test all these QTLs for interaction with our weather covariates. And we used this model that was developed by Marcos Malosetti, which fits a main environment effect, a QTL main effect, a QTL by environment effect, and the QTL by, a QTL by environmental covariate effect. And then there's a QTL by environment and the genotype effect, which are random effects here. And we could only find significant interactions for 2 of our QTLs in the indica data sets, and were both for head rise percentage. And the weather covariates were mainly related. First of all, we tried with all weather covariates, and the only ones we found significant interaction were with covariates that were during the maturity stage, which is reassuring. And these were weather covariates related to precipitation and humidity for one QTL, and for the other QTL related to temperature. And basically, the similar situation here in the tropical japonicas. We found one QTL for head rise percentage showed significant interaction with environmental covariates during maturity 2 related to precipitation and humidity. And for chalkiness, this QTL showed interaction with covariates that were related to minimum temperature and precipitation. And this was unexpected, because we know from previous studies that usually grain chalkiness in the tropics, at least, is mainly affected by high temperatures. So, but usually the high temperatures in the tropics, the ones that affect grain chalkiness, are those that are higher than 33 degrees. However, in Uruguay, temperatures, maximum temperatures that during grain filling stage, they never reach 33 degrees. They're always lower than that. So it could be possible that for chalkiness in Uruguay, and also maybe in other temperate or subtropical areas where temperatures are not as high as in the tropics, maybe there's other weather components that might be affecting this trait. It could be either minimum temperature or also like solar radiation. And there's some physiological studies within the Uruguayan germplasm that the breeders are doing in Uruguay that also point to this. So that was all about genotype by environment interaction. here we're moving to a different topic. What we tried to do was to select, to explore how to use genomics prediction to predict the best combinations of parents for breeding. And so what we try to do here was to, first of all, for the first objective was we try to see how many crosses in common we have when we select our crosses based only on the mean, which is easily predicted by, and the progeny mean, sorry, which is easily predicted by the mid-parent value, or by using a combination of both the mean and the variance in our progeny. And the variance is a little harder to predict. To predict the variance in our simulated progenies, we did simulations. And also, we try to evaluate different selection criteria for improving negatively correlated traits. So usually in rice, grain yield and milling quality are negatively correlated. So we wanted to see different ways to we can improve all our traits simultaneously. This was all done doing simulations, but also we have some of the crosses that were simulated here were actually performed in the greenhouse. So we tried to compare our predicted values. So these crosses, the progeny from those crosses was actually evaluated in the field in Uruguay. So we tried to compare the prediction accuracies that we had for those crosses with actual field data from the field. So this is a summary of what we did. We first started from a training population that consisted of 300 tropical japonica lines, elite breeding lines. These were planted in Uruguay, and we firstly performed 19 crosses between these members of this line, of this training population. We brought the F1 seed here to Cornell, and we planted it in the greenhouse. So these families were in the greenhouse, and we advanced them up to F4, and then we genotyped everything. Then we also performed 185 random crosses between these 19 families, trying to do like pairwise, trying to cross individuals from different families. And the progeny, the F1 from these crosses, was sent to Louisiana for double haploid development, where we could only obtain 43 double haploids out of 185 F1 seed here. And all these families were then sent to Uruguay for field evaluation. So this is just a description of our training population. And as I said, grain yield and milling quality tends to be negatively correlated with rice, and this wasn't the exception. So here you can see that grain yield is negatively correlated with both milling yield and head rice percentage, and it's also positively correlated with grain chalkiness. But what we actually want to do is to reduce chalkiness. So this positive correlation is undesirable. We use, so to make the 19 crosses here between members of this training population, we perform a cluster analysis here, so where we could detect 5 different groups in this population, and we try to cross individuals between different groups as a way to preserve the original diversity in the training population. And then we try to cross also individuals that were good for one trait with individuals that were good for the other trait. So these are the results. As I told you, we made, okay. As I told you, we made these crosses in the field in Uruguay. We brought the F1 seed here to Cornell. We planted in the greenhouse, and we genotype everything. So these are the results from the simulation for each trait. So we simulated all the possible combinations between these 19 families, and we calculated the mean and the variance of each simulated progeny. And here we can see the mean of each progeny versus the progeny variance here. So from these progenies, we try to select the top 100 individuals that were had either the highest mean, which is which are these red dots here, and I'm sorry, the reference here, I don't know, they got weird red. And then we try to select the top 100 individuals that had both where the top 100 progenies where with the maximum yield, with the maximum mean of the top 10 of the top 10% of the population. So this would be a combination of selecting based on both mean and variance. And in purple here, we can see the crosses in common between the two methods, which are depicted here. So here we can see that we have more crosses in common for grain yield than for the other traits. So we could maybe for grain yield, using just the mean of between the parents to predict our best process, maybe that could be a good option, and it's the easiest option. But also for the milling quality traits, we might want to evaluate whether using the mean between the parents or a combination of the mean and the variance in the progeny would be the best option, because we don't have so many crosses in common. And here, this is to show you the correlated response between the traits. So in the simulations too. So here we can see that usually, of course, grain yield and milling yield between these 19 families, they have a negative correlation. But between grain yield and head rise percentage, the correlation is not that strong. So and we can see that too here this is the distribution of the correlations between the traits across all the progenies. So even though this distribution is skewed to the negative side, we can see there's some progenies that still have a positive correlation. So there's still room for improvement here in this population. So what we tried to do here was to simulate, try to evaluate different selection schemes to improve both grain yield and quality simultaneously. So the first three selection schemes we used they differed on the selection on which trait we were applying the strongest selection pressure. So the first one was firstly selecting based on the 10%, top 10% lines based on yield, and from there selecting based on the other quality traits. And the second one was selecting first for head rise and then based on the other traits. And the third one was selecting first for chalkiness, then the other two. And then we also evaluated a multiplicative index where we consider all the traits simultaneously here. And here, this X is the value for that progeny, is the mean for that progeny for that trait. And this K here is the minimum value is the lowest value or the highest in the case of chalkiness for that trait across all progenies. And then we took the top 25 crosses from each that were selected from each selection scheme, and we calculated the mean of those progenies, sorry. And we subtracted that mean from we subtracted the mean in the training population from that mean to evaluate our response to selection. And so for selection scheme 1, we see that we had the highest response to selection for grain yield, but the response to selection for the other milling quality traits was very low. On the other hand, using selection scheme 2, we had a lower response for yield, but we had a higher response for milling quality traits. And also the second best scheme was the multiplicative index. So this could be something to have in mind in the future when we want to using these types of indices when we want to breed for these traits simultaneously. Then this is, so that was with the simulations, and this part is about the evaluations or double haploid line families in the field. So again, as I told you, we only could recover 43 double haploid families out of 185 F1C. So we didn't have the chance to try to choose which families to evaluate. We just sent everything, and everything was evaluated in Uruguay. And so we calculated the means for each family. And since the problem we had here was that these lines arrived too late to Uruguay, they arrived on December, so they were planted late, actually 3 months after the regular rice planting schedule. So for this reason, we think that grain yield was so low. If we compare it here, this is meaning for each trait in the training population as a reference. So yield was very low for this line. But on the other hand, for milling quality, for milling yield and head rise, the means for these traits were actually a little better than the training population. And this was surprising for us. The mean for grain chalkiness was very low compared to the training population. Was unexpected because these lines were during maturity. They were in maturity during the fall season, so temperatures can be very low for rice, but it's a good result because we want to minimize chalkiness. These are the trait correlations between these families, and the correlations are low, but again, since the correlations between yield and the other milling quality traits are actually non-significant. But again, as our plants, they were planted late and our yield was so low because of weather conditions, we can't take this very, these plants are, they're right now in the field. They were planted again for a new evaluation in the appropriate time. So we're waiting for that data to make more informed decisions about the results of this part of the analysis. So what we tried to do was to compare our prediction accuracies for these families with the actual data from the field. And so what we did, we performed genomic prediction on our lines. We had our prediction accuracies for the families. Then we had the evaluation from the field. We ranked both predictions and the field data, and we took the top 10%, and we compared which ones on the top 10% for the predictions were also in the top 10% for the field evaluations. And again, for grain yield, we could only find one family. This family was in common between the two rankings. But again, since they were planted late, we need actually like better data to examine the role of genomic selection for grain yield. But here for milling quality traits, we found 4 families out of 10 were in common between the two rankings, and for grain chalkiness, 6 families out of 10 were common in between the two rankings. So again, we are waiting for more data to analyze from the field, but this is promising for us because it could be genomic selection could be a good strategy to predict traits in the milling quality traits in rice. So that was everything about genomic prediction. Now this is a bonus track, and I'm gonna go over it a little fast here. But since I told you we could only obtain 43 double haploid families, actually a little more, they were 54, I think, double haploid families out of 185 F1C. So our double haploid yield was very low. So what we try to do was to explore our germplasm for anther-culture. Anther-culture is the process to obtain double haploid lines in rice. So we try to explore our germplasm for genomic regions associated to anther-culture in our tropical japonica lines. And mainly anther-culture consists of 2 main steps, which are callus induction and plant regeneration. And these are the 2 main bottlenecks of the process. So we know also that the response to anther-culture is highly dependent on the donor genotype, and usually indica lines are the least responsive among all lines. Tropical japonica varieties are easier to regenerate, but there's a big variation, a wide variation on response to anther-culture. So our objective was to identify QTLs for both callus induction and plant regeneration in our tropical japonica double haploid lines. So our data set consisted of 191 lines that belong to 54 double haploid families. These were all the double haploid families we could recover after the anther-culture process. And the traits were Cali transfer per dish. This is a measurement of callus induction. Number of plants regenerated per callus, which is a measurement of plant regeneration. And then the number of panicles per each regenerated plant. It gives us an idea of the vigor of our plants, because usually plants regenerate, but not usually, but sometimes plants regenerate, and then we transfer them to the greenhouse and they die, or they have very low yield. And then the genotyping was done using an external library, and we had a total of about 69,000 markers. So, I don't know why it looks so bad, but these are the results for our GWAS for callus induction. And here we could identify 4 QTLs that were distributed along chromosomes 2, 7, 11, and 12. Then for plants per callus, we could find a lot more QTLs. We found a total of 12 QTLs here. And some of these QTLs were mainly this one in chromosome 2 was also collocated with this one for callus induction. And these QTLs were also distributed along chromosomes 2, 3, 5, 6, 7, 8, and 9. And finally, for panicles per plant, we found a total of 5 QTLs. And this QTL also collocated with this second QTL found here for plants per callus. So we made a literature search to find which QTLs for these traits were previously reported in other studies, and we found a total of 8 QTLs among the ones we found were previously reported in other studies. So these results are exciting. Of course, this is the first step. We just identified the QTLs. Now we have to go more deeply into fine mapping and possibly identifying causal genes under these QTLs. But this is exciting. So that's about all I wanted to tell you today. This is the acknowledgement part. So now I want to first thank my committee, my advisors, Susan McCouch, and the other members of my committee, Jean-Luc and Philippe, and also Lucia, which is in Madison now. I also want to thank the breeders in the rice breeding program in India, which we could get all the data from them. So, and also the people from biotechnology and statistics, and also the current and former members of the McCouch lab, mostly Sandy and Cassie for taking care of my plants and helping me with the crosses, and Eugene and Francisco for helping me with all the for the bioinformatic analysis. And of course, Giovanni, Vishnu, Moni, who sadly left us too soon. Diane and Juan David for sharing of his time and conversation, etc. So thank you very much for being here.
This has been a production of Cornell University, on the web at cornell.edu.